All right, just a little advertisement for some express advising. So hopefully you're all thinking about signing up for next semester, next year. Need a little help? Drop in next week. All right, just a little blurb for that. All right, so I felt pretty good about the progress we made last lecture. So I don't feel quite so hurried today. So that's a good thing. I'll try to kind of, when we finish today, kind of say, okay, this is the plan. This is where we're going from here. This is what we're going to do. But remember, we do have an exam a week from today. So just a reminder. Okay, questions from last time before we move on? We had just started talking about adaptive immunity. This is chapter 17 of OpenStax. Talk about some differences between innate immunity and adaptive immunity, and also spend some time on talking about what antigens are. All right, antigens are, that name came from us calling them antibody generators. All right, so antigens are usually parts of some kind of pathogen that our immune system recognizes, and in response will make antibodies to target those antigens. So let's talk about antibodies, and then we're eventually going to get to how are antibodies made and what activates their process, as well as a cell-mediated response, which is a little bit different. Okay, so structure of antibodies. If you have some antibodies in solution and you throw some lytic enzymes in there to digest them, what you end up with is three fragments, two fragments that are identical, and then a third fragment. So you end up with these two, what we call FAB, or that stands for fragment of antibody binding. Sorry, fragment of antigen binding. Okay, so those two arms are the Fab fragments of the antibody. I should back up and say that we consider antibodies to be glycoproteins. They're mostly proteins. There's a little bit of carbohydrate associated with them, so that's where the glyco comes in. And we also sometimes refer to antibodies as immunoglobulins. Technically, antibodies are a kind of immunoglobulin, but oftentimes those two terms are just used to mean the same thing. Okay. There's two heavy chains here. I kind of skipped over that. So this part here and this part here, we call those the heavy chains because they have a higher molecular weight. And then there's two light chains, lighter molecular weight on the side of that. I mentioned the two arms, the Fab regions, and at the end of the arm, kind of very end here and the end here, that's the antibody binding site. That's where the antibody recognizes antigen. And so it's kind of like, one analogy may be kind of like a puzzle, like those antibody binding sites only fit a certain kind of antigen. And that becomes a little bit more obvious when you look at I think they call it like a space filling diagram, where Every atom is drawn in, so that's what this is. And you kind of start to appreciate, oh yeah, this is a really particular 3D shape, and that's only going to fit another particular 3D shape. So antibodies are extremely specific. They're only going to fit certain kind of antigen. There's a hinge region to the antibody, so that just means it's kind of flexible at that part. So like if you were standing here with your arms up, right, you're kind of flexible because we bend at our hips. And then this lower fragment, that's called the stem region. In other words, that FC, that stands for the fragment that is crystallizable. Maybe spelled that right, not sure. Okay. I often just call it the SEM or the FC region. And if you look at the gene fragment in the chromosome where the information to make the antibodies comes from, there's actually 5 different segments of that gene. So one's the mu region. We name those Greek letters, and you italicize gene segments. So the mu, the gamma, the alpha, the epsilon, the delta. When those, say when the mu region is transcribed, then that makes an IgM antibody. Okay, so these are the different isotypes of antibody. And again, that what isotype they are depends on that stem region. And that's really where the some of the function of the antibody is located. So let's, and by the way, just to be clear, these 2 regions where the antibody binds to the antigen, those recognize the exact same antigen. Okay, so you're not going to have one side recognizing one thing and one side recognizing something different. Okay, they're exactly the same. So, and we'll see why that's important, hopefully, as we go forward. It seems like it might be better if they recognize different things, but we'll talk about that. All right, so let's go through the different isotypes of antibody, and then we'll talk about what the antibodies actually do, how they help our adaptive immune response. The first kind of antibody that's always produced, so it's the B cells, remember, that are differentiating into plasma cells, and it's the plasma cells that make the antibody. The first kind that they start kicking out is always IgM antibody. So that's kind of what it's famous for. The first antibody produced in response to some kind of infection. It looks kind of unique because it's actually a pentamer when it's circulating in the blood. So there's 5 different IgMs that gets, how do they coalesce into that structure? There's something called a J chain, which is this little, that's right here, that little arch there. It's a piece of protein. It has something to do with assembling those 5 individual IgM monomers into a pentamer. I won't go through the mechanics. I have no idea how that actually happens. We call that J chain an accessory molecule. And again, it has some role in joining them together. Okay, this is actually not a lot of, when you look at the total antibody in circulation and the amounts of each isotype, it's not a lot of the total antibody in the blood because this kind of antibody doesn't last for very long. And then those plasma cells will change into the kind of antibody that's best suited to fight that whatever that infection is. Okay, but it's got, so there's 10 binding sites here. And again, all those binding sites are recognizing the same antigen. Okay, so this kind of antibody is going to be really good at kind of clumping a lot of different antigen together. So we'll talk about that more in a minute. Question? No, it's got, so each antibody like this is a binding site and this is a binding site, right? Like each one has two. Yep. So 10 binding sites for that IgM right there. Yep. So it's a lot. Okay. So, like I said, the IgM, yeah, another question. Oh, sorry. Go ahead. No, let me erase what I scribbled over here. So where's the J chain? So this little right here, can you see that? Yeah, that's the J chain. Yeah, it's hard to appreciate how that actually happens. There must be some other bonds in there that assemble those, but we're not gonna worry about that. Okay, so so that we have a plasma cell that's secreting these enzymes. At some point, it will get some cytokine signals and it will change the antibody that it's producing. So we call that class switching. Okay, when, due to cytokine signals, these plasma cells will switch to the kind of antibody that maybe is best going to fight an infection. Even though it switches the stem region, the specificity of the antibody stays the same. Okay, so that antigen binding region doesn't change. Most often, it's the plasma cells are going to switch to start making IgG. Because that's the most common antibody in circulation. About 80% of the antibody in the bloodstream is going to be IgG. It's the longest acting antibody. So it can persist for sometimes months in the bloodstream. And what IgG is famous for is that it's the only antibody that can cross the placenta. Okay, so that's really important to protect the developing fetus. Some of the mother's antibodies will cross the placenta, and then when the baby's born, those antibodies will keep working for a number of months after it's born. So the antibody has at least some of its mother's protection still. What's the other thing I was going to say about that? Oh, so babies that are born prematurely kind of miss out on some of those antibodies that they would get from the mother. Because it's the only one that crosses the placenta, the one problem that can be encountered is in Rh disease. So everybody either has or doesn't have that rhesus antigen. So if you're a blood type, say A positive, you're positive for the rhesus antigen. If you're A negative, you do not have that rhesus antigen. So that can become a problem if the mother does not have the rhesus antigen, but her developing baby does because the father had it. Okay, so usually that's not a problem during the first pregnancy because maternal and baby's blood probably don't mix very much during the first, 9 months, but during birth, they're probably going to mix, and that can sensitize the mother for the second pregnancy. She can start making antibodies against that rhesus antigen if the baby has it. And that can cause, at best, some anemia for the newborn. At worst, it can cause a hemolytic disease of newborns, and it can kill the baby. So, and again, it's all because of these IgG antibodies. Now we have RhoGAM shots, and with good prenatal care, the mother's going to get that to desensitize. So she kind of masks those antibodies that she's producing. So it's not today. It's not a problem. We came out with that in like the 1960s, I think. Okay. Another way that newborn babies, though, can be protected and to get some maternal antibodies, sometimes there's some passing of IgA antibodies because this is the main antibody present in mucous membranes and bodily secretions. So by bodily secretions, I mean things like tears, saliva, and breast milk. Okay, so it's actually a pretty low amount of antibody in the blood itself, but is the most prevalent antibody on our mucous membranes. Okay, and when I believe it's a monomer in the bloodstream, but when it's secreted onto those mucous membranes, again, we get a J chain, which is over here, that joins those two monomers to make a dimer. Okay, so now we've got 4 antigen binding sites. And there's another component here, this secretory component that's drawn in over here, that enables the antibody to be somehow, secreted onto those mucous membranes, and it has some protective effect from our proteolytic enzymes. So in our mucous membranes of our digestive tract, for example, there's lots of enzymes that are mixing around in there to digest our food. We don't want those enzymes to kind of non-specifically break down our antibodies that are there to help guard against foodborne pathogens. And that's especially important because if infants are going to reap any benefit from those IgA antibodies in their breast milk, we want the infants' bodies to digest the antibody as well in their digestive system. So that secretory component, again, protects it a little bit and makes it a little bit more longer acting. All right, and then IgE and IgD. IgE is very rare, typically less than 1% in the blood. We do see more of it though if the patient is fighting a certain kind of infection, like some kind of infection by parasitic helminth worms, or during some kind of allergic reaction. Okay, so we get, again, more IgE binding to basophils and eosinophils. Remember, those were the kinds of granulocytes that act on eukaryotic pathogens. They have receptors for IgE, and the IgE will cause them to degranulate. Okay. IgD, I don't have much to say about IgD. We don't know a lot about it. It appears to act as a B cell receptor. We're going to talk more about B cell receptors shortly, but we don't think that they are actually secreted ever from the B cell. Okay, so instead of the cell pumping out lots of antibody and releasing it, the IgD just stays bound to the B cell. So there's actually several different kinds of B cell receptors, and this is one of them. That's a great question. So do we understand why patients get IgE flare-ups if they're getting another kind of antibody, like passively, like by IV or something? I really, to be honest, don't know anything about that. There must be some kind of cytokine signals there that cause some kind of cross reactivation or something like that, cross stimulation. That's a great question. Okay, what do the antibodies, what do they actually do? So 5 different isotypes, and by the way, there's other isotypes and other kinds of animals. I think sharks have IgW, for example. So those are the ones that humans have. So 5 different functions of antibodies, and certain isotypes are going to be better at certain functions. I'm not too worried about you knowing exactly which function each isotype does. But one very common function is one of neutralization. So in essence, the antibody is going to block some kind of adhesion molecule of pathogens. Okay, so it could be a bacteria, it could be a virus, maybe even a toxin. Okay, so here it shows some antibody binding to the viral spikes. Remember, the spikes are going to be important to attach to host cell receptors. If all those spikes are covered up, then the virus can't infect a certain cell. Here it's showing some antibody binding to a toxin, right? So then the toxin can't act where it's supposed to. That's all covered up. I'm not sure why they show antibody, if they're illustrating adhesion molecules, they show, it looks like a flagella, antibody binding to a flagella. Flagella is not adhesion, right? That's more motility, but okay, that's possible too, all right? Activation of complements and inflammation. So we talked about one of those activation mechanisms of complement pathways involves an antibody bound to some antigen, and then the C1 recognizes that and starts in motion that whole complement cascade. Okay, so that's in the what we call the classical activation pathway. And then we end up with, increased inflammation and chemotaxis of other white blood cells. Some of those complement fragments act as opsonins, remember, so they're going to bind to pathogens and make phagocytosis more likely to happen. It can also result in a membrane attack complex and poking a hole in some kind of pathogen, usually gram-negative. Or an antibody can act directly as an opsonin. Okay, so remember that we'll say stimulates phagocytosis. Phagocytic cells, like say a macrophage, are going to have receptors outside of their cell for that stem portion, the Fc portion of the antibody. And so if there are antibodies coating the pathogen here, then that's just a really nice handle for phagocytic cell to grab a hold of. Even if there's a capsule around that pathogen or some other anti-phagocytic virulence factor, the macrophage is going to be a lot more successful in phagocytizing it. This was the one that I mentioned before, agglutination. Basically, antibody can make the pathogen stick together in clumps. Okay, our immune system is much more successful in recognizing things that are larger. We said that big molecules make better antigens than small molecules. So this is where it becomes important that all those binding sites recognize the same thing. If they recognize different things, then they wouldn't be very good at clumping things together. So you can make big clumps of pathogen. That's going to be noticed really easy, whether it's in the lymph, it's going to be screened out at the lymph nodes, or it's in the bloodstream, it's going to be filtered out by the spleen. We'll talk more about anatomy of lymphatic system, should get to it today. Okay, so again, the IgM is going to be really good at that because there's a lot more binding sites with agglutination. And lastly, this antibody-dependent cellular cytotoxicity. This is going to be facilitated by NK cells, natural killer cells. Okay, so basically, these natural killer cells also have Fc receptors, and if an antibody is bound to an antigen and it recognizes that Fc portion, then that will activate the NK cell to release some cytotoxic cytokines, the same ones that are released by cytotoxic T cells that we'll talk about later, that can act on the pathogen and kill it directly. Okay, so the antibody can mediate the killing of pathogen by natural killer cells. Right. So, and then there's a table in your book that kind of sums up the structure of all those isotypes and kind of, you know, relatively how much relative percentage, some other things, function. Again, you don't have to worry about which isotype is best at which kind of function, but it's kind of nice to just kind of see everything in one place there. Okay, all right, so I'm kind of going out of the order of the book just a little bit because it just seemed like it made more sense. Okay, so don't worry. I next want to talk about, so I said that the antibodies are made by B cells. So let's talk a little bit more about B cells next, and then we'll get into our T cell discussion. So B cells, they are made in the red bone marrow. I have another slide that talks about this again later. Red bone marrow is not found in all of our bones. It's mostly the kind of larger, flatter ones. So like the head of the femur, in your hip, shoulder blade, those are places in our body that has red bone marrow. And the B cells, again, are going to be made in that red bone marrow, and they're also going to mature there. So, so I'll write that, even though I have it written later. Made in, so B cells made in red bone marrow. And we'll say, and they also mature there. What do I mean by mature? That's where they're going to kind of develop their B cell receptors, which we're going to talk about next. And it's also where the B cell receptors are going to learn to not recognize any autoantigens. Remember, autoantigens are any antigens that we have on our host cells. We don't want our immune cells recognizing host antigens, because then they're going to have just inappropriate response. So we don't really go through that whole selection process. Just mention that it takes place and that there are mechanisms so that our B and T cells don't recognize ourselves. Obviously, that's important. And hopefully nothing goes wrong with that, or else we have autoimmune disease. All right, so if we think about what the B cell receptor looks like, if you look at it, it should look kind of familiar because it looks just like an antibody, which we just talked about. So there's two heavy chains, there's two light chains, there's these antigen binding sites. And this is going to be anchored right in that B cell plasma membrane. Okay, there's one drawn here, but there's actually somewhere in the order of 100,000 B cell receptors on every B cell. Okay, so that's a crowded place on the outside of a B cell, lots of B cell receptors. And they all have, all of those on that one B cell have the same specificity. And again, it seems like, wouldn't it make more sense to have a B cell that had lots of B cell receptors that could recognize 100,000 different things, rather than only have a few B cells that have a certain B cell receptor, and then those B cells have to find, you know, or come in contact with the pathogen. Well, actually, when a B cell does recognize the epitope of an antigen, that sends a very weak signal into the B cell. So actually, lots of receptors have to recognize the same thing to send a strong enough signal before the B cell can act. And it often takes other signals besides that as well. Okay, so same thing, both variable regions are going to recognize that same antigen. Okay. So the thing that kind of bothered researchers for a really long time is that they knew that we had B cell receptors that recognized almost anything that we might ever come in contact with. Again, besides our self antigens, hopefully. But if you follow that, usually there's, one gene, one protein theory, that would make a whole lot of genes that we have. Do you know about how many genes that we have in our body? Yeah, about 20,000. That's the number that I've heard too. So we know that we have B cell receptors that can recognize millions and millions of things. So how is this possible? We don't have a gene for every B cell receptor. Well, they figured out in the 1970s, some Japanese researchers figured out that it's not one gene to one B cell receptor. Okay, so that is certainly not the case. What we think happens, that we call this BDJ recombination. Okay, so which is actually a very complicated process, and this looks sort of complicated, but basically on the gene where the heavy chain, we know the instructions for the heavy gene come from, and then it's transcribed and it's translated, that protein. The gene actually has several variable regions. So it has one variable region that has 65 different possible V regions, and then there's 27 D regions and 6 J regions. So what they think how this recombination works is that a protein, here they've called it RAG protein, it randomly goes through and picks out one V region, one D region, and one J region. And those get transcribed and translated to make both the heavy chain and the light chain. Okay, so think about it. It's like you get up in the morning, you have 65 shirts, you pick one at random, you pick randomly one of 27 pairs of pants and one of 6 pairs of shoes. Okay, and then that's your outfit. Now, the problem is sometimes that outfit doesn't match and you have to go back and change into something else. Well, sometimes when that RAG protein picks each one of those, that doesn't make a protein that's maybe translatable. So not all of those actually make it into the actual B cell receptor. But they've done calculations, and you don't need to worry about these calculations, but taking into account all the possibilities of, again, these different variable regions and some other recombination events, and considering both the light chain and the heavy chain goes through this, they've come up with this number down here that we have 10 to the 23 possible B cell receptors. So that's how that enormous variability, we think, gets generated in those B cell receptors. Okay, so again, we have a B cell receptor that would recognize pretty much anything that we would come in contact with, only a few cells though. And then once those get activated, they will make more, that clonality, make more of them, they proliferate. Okay, so a little bit more about B cells, and then we're gonna move on to something else and then come back to them. They actually, so when they get activated, right, well, we'll come to that in a minute. They get activated in 2 different ways. Sometimes they're going to be activated, I don't need to write that again, by antigens that need T cell stimulation. So we call these T-dependent antigens. And sometimes they get activated in a different way by antigens that don't need T cell stimulation. And as you would guess, we call these T-independent antigens. Okay, so we're going to go through both those scenarios probably later on in this lecture if we get to it. So we'll come back to that because we need to talk about T cells first before we get to that. But once the B cells get activated, whichever kind of antigen it is that activates them, what happens? The B cells, they get bigger, they start to rapidly divide and differentiate into these plasma cells. Okay, remember the plasma cells are the ones that are secreting the antibodies. And sometimes, it depends how it's activated, but sometimes we get memory cells as well. So basically a memory cell, the next time that memory cell encounters that pathogen, it doesn't have to be activated all over again. It's ready to go, and it's ready to start kicking out, very quickly differentiate into plasma cells and start making antibodies. This is one of the coolest pictures that I really appreciate this picture. This is a plasma cell of a guinea pig. And you know, when you get pictures of eukaryotic cells, you know, usually ones they show you in textbooks are, oh, here's the lysosome and here's the ribosome. You know, there's like one of everything. It's not really the way it is in real cells. It depends what kind of cell that is. So in this cell, you can, you know, the big brown spot there, that's the nucleus. And I think the bluish blobs are mitochondria. Everything else packed in that cell is rough endoplasmic reticulum. Okay, so those ribosomes that are studied on that rough ER, those are making proteins that are destined to be secreted outside of the cell. Okay, so you can tell from looking at this cell that its job is to secrete some kind of protein. Okay, so activated plasma cells kick out about 2,000 antibodies per second, which is really mind-boggling. Okay, so really fast metabolism. They typically don't live very long. They just kind of, they burn out because they're, again, producing so much protein so fast. Now, there are different kinds of plasma cells. There are ones that are more, that are kind of slower, and then they live longer. But I won't get too much into that. All right. So we talked about B cells, and we talked about antibodies and how antibodies work. Like I said, we'll circle back to how those B cells get activated, because we don't want to be making a bunch of antibodies if it's not appropriate to do that. We don't want to waste all that energy in making all that protein. So we need to talk about T cells first. And to understand T cells, we have to first have a discussion of MHC glycoproteins. And I've alluded to these already a little bit. This is the mechanism by which many different kinds of cells will present antigen to T cells. Okay. So MHC stands for major histocompatibility proteins. And they're coded for by genes called the major histocompatibility complex. I mention that because these genes are polymorphic. And that means that among us humans, for example, we all, most of us have very different major histocompatibility genes. Okay, so like you're more likely to have similar genes to someone that you're related to, but to someone on the street, you might be a match with like 1 out of 500,000 people. Okay, so there's a lot of variability in that gene segment. So that's one of the things that they look at. If they're looking at organ donors, they try to match up the MHC genes with the recipient and the organ donor, and then there's less chance that that organ will be rejected by the recipient. Okay, and that also plays into why, you know, maybe 2 of us could be exposed to the same pathogen at the same time, the same amount, and one of us gets sick and one of us doesn't, because there's differences in how that pathogen sort of gets presented to our immune system. Okay, in humans, they have another name for this called HLA. You might read about that, human leukocyte antigen. Let's see. Notes are all out of order here. Yeah, human leukocyte antigen. So I don't know why they gave it a different name in humans, or why don't we just call it HLA all the time? But I don't know. That's just referring to MHC glycoproteins in humans. Okay, and like I said, they're going to hold epitopes for presentation to T cells. Right, there's two different kinds of these membrane spanning proteins. MHC1 is present on all body cells except red blood cells. Okay, so basically they're present on all body cells with a nucleus. And red blood cells had a nucleus and then they lose it because apparently they don't need it anymore. So without that nucleus, they're not able apparently to make these MHC glycoproteins kind of constantly kick them out. And MHC1 are going to present endogenous antigen. Remember, that's antigen from within a cell, usually antigen, say, from a virus. And it's going to present endogenous antigen to cytotoxic T cells. Okay, I know we haven't talked about that. We're going to talk about that segment of cell and what they do shortly. And then MHC class 2 are only present on certain kinds of cells. They're present on antigen presenting cells. And those are the macrophage, the dendritic cells, as well as B cells. So B cells are not exactly phagocytic. They have kind of a different mechanism to ingest antigen and then present it with MHC class 2. So macrophages and dendritic cells are phagocytic. Another kind of cell that is phagocytic are the neutrophils, but they don't really present antigen because they don't live long enough to do that. Okay, so the macrophage and the dendritic cells are going to be traveling throughout the body and the lymph, presenting antigen. Okay, so that's why it's not the neutrophils. And this class of cells, they're presenting exogenous antigen. So that's antigen that they have phagocytized and now are presenting a piece of that, and they're presenting that to T helper cells. All right. So it's always good to have a picture. What's going on here? I'm not so worried about the structure of these. So this is a little bit more detailed, but the MHC class 1, it spans that the membrane, remember, of all of our cells, all nucleated body cells. It spans it once. So basically all our cells have this as a mechanism to kind of advertise what's going on within the cell. Okay, so we're going to look at kind of a basic graphic of how that works, but basically every cell will constantly sample kind of what's going on outside of the cell. So I like to think MHC class 1, I'm raising my hand, I'm volunteering what's going on inside the cell. Okay, so that's MHC class 1. And assuming that it's just regular cellular proteins that are being presented, those get ignored by other cells of our immune system. Those are autoantigens and those should be ignored. Okay, so let me write this. Volunteering, we'll just say amino acid bits of cellular activities. Okay. Ignored normally. Okay. But again, if that cell is infected with a virus, this is how the cell advertises that what's going on. Okay, I need some help. I got a virus in there or some other intracellular pathogen. Okay. And then the MHC class 2, macrophages, dendritic cells, and B cells, remember, they also have MHC class 1 because they got a nucleus. So they've got both MHC1 and MHC class 2. And they are, MHC class 2 are presenting amino acid, let's say fragments, that sounds more scientific, that were phagocytized. Okay, so if MHC class 1 is raising your hand and volunteering, MHC class 2, you're handing somebody something with 2 hands, and it's like, here, this is what I'm dealing with. Okay, the macrophage is traveling through the lymph looking for a T cell that it can, get some help with what it has phagocytized. That's kind of the way my brain works and helps me remember. Okay, so kind of a brief, sort of a basic idea of how that works. Again, this is more complicated than what we'll present it, but with the MHC class 1, there are proteasome complexes in the cell, this complex of enzymes that goes around and kind of chews up bits of the cell. And you would think that might be bad for the cell, but apparently it's not. It all works out. It just samples bits of the cell. And so that's where it says here, the polypeptide is catabolized to some protein within the cell. And that enters into the endoplasmic reticulum. And that's where the MHC class 1 are expressed inside endoplasmic reticulum. And those epitopes, maybe 10 to 20 amino acids long, bits of proteins, enter into the ER, and then, may or may not match up with the specificity of those MHC glycoproteins. Okay, that pinches off into a membrane-bound vesicle, and then moves to the surface of the cell, and then are expressed on the surface of the cell for a time. Okay, after a while, those degrade and new ones are expressed. I don't know the timing of that, but that's kind of just the basic idea, again, of how those that advertisement takes place, that normally what's going on in the cell. Okay, the MHC class 2, remember, here's, say, some kind of phagocytic cell here, and it's phagocytizing some antigen. It doesn't show this, but the MHC class 2, again, these originated in the ER. It pinches off from the ER and fuses with the phagolysosome. So remember, the bacteria, say, gets enclosed in a phagosome. When that membrane fuses with a lysosome, then we call it the phagolysosome. The enzymes of the lysosome will hopefully chew up that pathogen. And so again, we get these kind of bits of pathogen. Maybe some of them, match up with the MHC class 2, and those, again, that endosome would move to the outside of the cell, and then the phagocyte is presenting that. And again, this is kind of moving through the lymph. We'll get to kind of where that's happening shortly. Okay. And again, depending on what MHC you have, maybe you recognize that bit and it gets presented and you have a very quick response to whatever pathogen you've encountered. If it doesn't match up quite so well with your MHC, maybe your response is going to be a little slower. All right, questions so far? Okay. So putting that together, I already mentioned cytotoxic T cells and helper T cells. Let's go through kind of what their job is, and then look at T cell receptors, and then we'll kind of put everything together. So what cytotoxic T cells do, they are going to kill cells that are infected with some intracellular pathogen. Okay, so host cells. Okay, there's a homework question, you got to read really carefully. But so cytotoxic T cells, their job is to kill our cells that have been infected. So again, there's lots of checks and balances on these. We want to make sure that those cells are not acting inappropriately. We think about, if you use the whole ammunition analogy, antibodies are like bullets, like they're cheap. A cytotoxic T cell is more like, you know, a smart bomb or a tank or something that's going to do a ton of damage. So we have to make sure, again, that it's stimulated appropriately and it doesn't act unless it has the right stimulation. Okay, so it's killing our cells that have been infected. They have a CD8 glycoprotein on the surface of them. And that's kind of how we identify them. They're CD8 positive. That CD stands for cluster of differentiation. Okay, and that actually binds to the MHC class 1. Okay, we'll show you a picture of that in a minute. So cytotoxic T cells, I might have mentioned this before, but they recognize antigen presented by MHC class 1 only. Okay, that's the ones, that's the MHC class 1 is advertising the intracellular pathogens. Okay, so we'll talk about how those are activated. We'll get to that, and what they actually do when they're activated. How do they kill our host cells? Helper T cells, they are sort of, I think I said they're the general in charge of the entire immune response. They have a CD4 glycoprotein on the surface, so that's a very famous subset of cells, helper T cells, CD4 positive T cells, because those are the ones that are killed by HIV. And T helper cells are going to recognize antigen presented by MHC class 2. Okay, we're gonna, at a further detail, there are different subsets of T helper cells. Again, they differentiate by getting the correct cytokine signal. But it's Th1 cells that are going to help stimulate the cytotoxic T cells, and it's the Th2 cells that are going to play a role in activating the B cells in order to differentiate into plasma cells and make antibody. Okay, so we'll get to that. There's a third type of T cell that we're not going to talk about anymore. Just it gets really complicated how they work, but there are some regulatory T cells, or sometimes called Tregs, that just make sure that the cell-mediated response is not kind of over the top. So there's some kind of regulatory function there, but we're not going to talk about those anymore. All right, the T cell receptor are around, there's thousands of them. I don't have a particular number like I did for the B cell receptor, but there's thousands on each T cell. Okay, and that's what's pictured here. Just to kind of compare to the B cell receptor, I put that down there in the corner. There's some similarities. They're both, anchored in the plasma membrane. They both have an antigen binding site at the tip of them. They're both very specific, right? So the T cell receptors, we think, are made with that whole VDJ recombination, just like the B cell receptors. Okay, so very specific. The main difference here that we've alluded to already, the B cell receptors recognize epitopes of antigen directly, where the T cell receptors, they only recognize antigen that's presented to them by MHC glycoproteins. Okay, so they're fussy. You have to show them the antigen in just the right way, otherwise they won't look at it. All right. All right, so before we move to think about how that activation will take place for the T cells and the B cell, there's one other piece that I want to talk about, and that's a little anatomy. So I actually never had an anatomy class, so if I say something ridiculous, just let me know. But we want to think about like where these reactions are taking place and kind of the important aspects of our lymphatic system, right? So I already mentioned a primary lymphoid organ. Our primary lymphoid organs are where lymphocytes, B cells and T cells, are made. Okay, so I already mentioned this before. Sorry about repeating myself. Red bone marrow. We talked about how that's where B cells are made. That's where B and T cells are made. And it's where B cells mature. So we talked about that. The other organ that's considered a primary lymphoid organ is the thymus. And you guessed it, that's where T cells are going to mature. And I assume that's where the name came from, T cells, because they mature in the thymus. So this is a little organ. It's kind of small here. The picture is small there, but it's a little organ that kind of sits on the top around the heart. And it does most of its work actually before we're born. And then as we age into adulthood, it kind of shrinks up and really doesn't do much anymore. So basically, we have a mechanism. T cells, once they're made in the red bone marrow, they travel to the thymus, and there's a mechanism where they are bombarded with all sorts of possible antigens that they could come in contact with. And if it's a self antigen, like pieces of our kidney is presented and from all parts of our body, if it's a self antigen, those T cells with those T cell receptors are deleted, okay? Because we don't want T cells that recognize autoantigens. So there's a whole mechanism to do that, and that takes that sorting process takes place in the thymus. Again, mostly before we're born or when we're very young. And then the secondary lymphoid organs, this is where B cells and T cells become activated. Okay, so this is where B and T cells come in contact with antigen. Okay, so the first thing that we think of are the lymph nodes. So we have somewhere between 500 and 700 lymph nodes in our body. They're about the size of a pea. So we've got plenty of them. You can lose some of them and it doesn't do much difference. But they're concentrated in the groin, the armpits, and the neck area. And that's where all these lymphatic vessels, we'll get a close up of those in a minute, where the fluid is traveling through lymphatic vessels and it has to kind of mix in these lymph nodes periodically. Okay, so filters lymph. There's lots of lymphocytes in the lymph, right? That's where B and T cells mostly are. And that's where macrophages and dendritic cells, our antigen presenting cells, are coming in contact with the B and T cells. Other secondary lymphoid organs, the spleen. The function of the spleen is to filter the blood. So the lymph nodes filter the lymph and spleen filters blood. And it's, like I said, it's most likely going to notice larger antigens, ones that are all clumped together. We can survive fine without a spleen. So you might know somebody that's been in an accident and the spleen ruptured. Apparently you can't just sew a spleen together. Like, you will bleed out if the spleen ruptures. So they just have to remove it. People that don't have a spleen are a little bit more susceptible to certain kinds of infections, mostly bacterial infections. So they might need more vaccines than typical people, or they may need to take antibiotics prophylactically if they know they've been exposed to somebody that's been ill. Okay, of course, the tonsils, a lot of us get along fine without tonsils. They're basically lymph nodes in the back of our throat that have kind of a hard outer covering. You know, why do some people need to get their tonsils out? Okay, they used to do this like to most kids, like 50 or 60 years ago. Most kids, they just took their tonsils out when they were little. Some people, they're fine with their tonsils. Some people, especially if they're large, they're particularly grooved, and you know, bacteria can get caught in them, and you can end up, you know, having especially strep throat more often. So yeah, some people are just better without it. And another kind of secondary lymphoid, we kind of call it an organ, is the MALT. And that stands for mucosa associated lymphoid tissue. So this is basically tissue. It's similar to lymph nodes, but it doesn't have this hard outer covering. I think the appendix is considered part of the MALT. And there's this other specialized tissue called Peyer's patches, and that's MALT tissue in the intestine. It's very important because to try to kind of sift through what's passing through our intestine and try to find pathogens that we've ingested. So anything that would give us food poisoning, we would call it, that's the Peyer's patches job, to kind of sense that and build up an immune reaction to that. All right, so we look more closely, the picture shows lots of lymphatic vessels. So, right, we don't have a lymph pump, right? How does the lymph move through those vessels? Well, the lymph moves in one direction, and it empties out in the subclavian vein. So that's, I don't know, up here by your collarbone somewhere. And that's the part of, that's the vein where the blood pressure is the lowest. Okay, so where the blood pressure is the lowest, the lymph will dump back into the bloodstream. And everywhere else in our body, basically the fluid that is kind of seeping out of our veins a little bit at a time gets collected by the lymph, and then it eventually dumps right back into the bloodstream. Okay, so kind of a close-up of, oh, I forgot. This is just a close-up of the thymus. Okay, there you have it. This is a close-up of a lymphatic capillary. Okay, so you can see the lymphatic capillary is in green, and then we've got a blood vessel to the left of it there. Of course, our blood is pumping in one direction, and our lymph only moves in one direction as well, because there are these valves here that keep it only going one direction. We don't want it just squishing around wherever. Okay, so when our blood is pumping through our blood vessels, there's going to be fluid coming out of there a little bit. There's going to be, of course, gases being exchanged, oxygen, CO2, and there's also going to be some cells leaving. Not red blood cells, they're not ever supposed to leave, we'll say white blood cells. Okay, so even more during inflammation, if you have more cells leaving, remember we called that process extravasation, okay? When those blood vessels dilate and the gaps between the cells get bigger, okay, and the cells are able to leave. Okay, so all of this sort of gets collected out here in the tissues, and when the pressure in the tissues is greater than the lymphatic capillaries, then that all that stuff will enter into the lymph. Okay, how does lymph move? Just by smooth muscle contraction. Okay. Apparently, when we are active, maybe we're exercising, that might skeletal muscles might move the lymph a little bit more too. So that's another reason to be active. So it's moving it rather slowly through those lymphatic vessels. Okay, and eventually it will come in contact with a lymph node. Okay, and you can see here the lymph node. There are several afferent lymphatic vessels. It's drawn 4 different vessels coming into the lymph. That prefix A means towards. Okay, so towards the lymph node. And there's only 2 efferent ones, okay, away from the lymph node. So that creates kind of a bottleneck in the lymph node. It creates, you know, some mixing around of that lymph. There's lots of cells in there, B and T cells that are now having the chance to come in contact with the antigen presenting cells like the macrophages and the dendritic cells. Okay, so this is mostly where those B cells and T cells are going to become activated. If there's an unusually large number of cells there and an unusually large amount of fluid, then what happens? The lymph nodes are going to swell up. Okay, so that's often a sign that the body's fighting some infection if your lymph nodes are swell up. So the doctor might feel for those, especially around your throat. And as that infection gets cleared up, then the swelling will gradually go down. Okay, so again, this is where all of the activation that we're going to talk about next is probably taking place. Okay, so are you still with me? I know, I feel like it's just a lot of stuff for you this lecture. We're doing good. Okay, so let's start with B cells and how they get activated. I said that there's 2 different ways that they can be activated. The T cell independent antigens, let's talk about them first because that's a little bit more straightforward. Okay, so remember T cell independent, that means that the B cells get activated just by recognizing a certain antigen. It doesn't need extra signals from a T helper cell. What kind of antigens? This is not the most common kind. We'll say this here, less common. What kind of antigens can stimulate B cells directly? Antigens that are large and that are not proteins. So most commonly certain polysaccharides, most commonly like bacterial capsules. And it doesn't appear to be all bacterial capsules, certain ones, okay? And so there's actually, I kind of wrote this kind of small, there is actually a second signal that has to take place, not from a T helper cell, but B cells actually also have those toll-like receptors, like what we talked about that macrophages and dendritic cells have, in order to recognize those PAMPs, those pathogen-associated molecular patterns. So there is a little second signal that B cells get from recognizing some broad-based pattern. What's good about this kind of B cell activation? It's a pretty fast response, faster than, as we'll see, the other way that they get activated. Still takes a little bit of time to make antibody. You know, it's not kind of instantaneous. So it is fast response, and that's good. The problem is it's not quite as good a response because there's no memory cells made. And again, the advantage of memory cells is that as soon as they recognize the pathogen, they will start making antibody right away. They don't need other signals from other cells. So it is pretty fast already, but not as fast as what memory cells would have. And there's no class switching. So in other words, we get only IgM antibodies. It won't switch to IgG or IgE if that would better fight the pathogen. Okay, so kind of one response. and no memory. And the other big problem is for a certain population of people, we don't get a very good response. We don't get much of any antibody made in young children. So how young? Say less than about 4 years old. I've read like different ages there, but we'll say about 4. Okay, so why do little kids not have a very good response? Probably their B cells are just immature. Okay, so for whatever reason, a young, a 2-year-old child wouldn't really make antibody at all to these pathogens. So they're particularly susceptible to pathogens that have T cell independent antigens. So one example of that, something maybe you haven't heard of, it's a bacteria called Haemophilus influenza. It's why little kids get the Hib vaccine. That's one of the earliest ones that they get. Stands for Haemophilus influenza B. And this is a pathogen that doesn't cause flu. We thought it did, and that's why we named it that back in like 100 years ago. It causes bacterial meningitis. So a very serious infection that adults don't get because we have a good response to that and we fight it off. Young children don't. So that's why it's really important to get them vaccinated, even though it's something that, as adults, we don't get and may not think about it as being important. Okay, so that's just one example. Talk about some other examples of T-independent antigens as we go forward. But here's your book's graphic explaining that. So here we have a B cell that's recognizing some bacterial cell. Looks like maybe it's the capsule. Oh yeah, polysaccharide capsule. And then that B cell, it doesn't show it differentiating into plasma cells, but then the plasma cell is kicking out these IgM antibodies. Okay, and remember, no memory cells. Okay, so good. I think we have time to talk about the other way that B cells are activated. I know that you're getting tired of hearing my voice. I understand. T cell independent antigens. Okay, these are the most common way that B cells are activated. And all protein antigens need this additional T helper cell stimulation in order to activate B cells. Okay, so I tried to kind of write on here the steps of it to refer to, but let's, so you can go back to that. Essentially, here's your book diagram. And again, they really simplified this quite a bit. So you may have some questions like, oh, how does that work? But I'll try to walk through it here, and maybe we'll walk through it again at the beginning, or I think what I'll do is post a short lecture. Hopefully I'm going to do it tonight, finishing up adaptive immunity, and then watch that before Monday and let me know if you have any questions. But I don't want to present new material right away on Monday, right? Two days before the exam. So, you know what, maybe instead of, no, I'll walk through it once and then I'll do it again. Okay, so give me a few more minutes. All right, so here we have a B cell that's recognizing some pathogen. So it has a B cell receptor that recognizes epitopes of that pathogen. And then what they don't really show over here, like before it gets to this step right here, that helper T cell actually has to become activated first. So say you have a dendritic cell. So I'm going to draw it like this. And then dendritic cell has ingested, phagocytized some pathogen, and it's expressing it, presenting it with MHC class 2. Here's some epitope. And then you have a T helper cell that with its T cell receptor recognizes that same antigen. Here's the CD4 that recognizes the MHC class 2. Okay, and now when it's made it's got that T cell receptor that recognizes the antigen, now that T helper cell will kind of self-stimulate or maybe some cytokines secreted by the dendritic cell as well. It will differentiate into T helper 2 cells. Okay, so this helper T cell has actually already been activated. And when that T helper 2 cell recognizes antigen that the B cell here has actually ingested a little bit of this antigen and presented it by MHC class 2. Now, this T helper 2 cell releases the cytokines that will stimulate the B cell to become activated. And when it's activated, we call it clonal expansion. It will make much more copies of itself. It differentiates into plasma cells that make antibodies and also some memory cells. Okay, so we got a long way. Like I said, look for a shorter lecture that I'll post hopefully tonight. Watch that before Monday. We'll do some reviewing, but then the exam will cover through that little lecture that I'm going to post tonight. Okay, thank you for coming.
Professor, how's the immunity class or the immune